Thank you so much to the amazing organizers here for the opportunity to share today. This is primarily going to be a project about ecology in California, and so I really appreciate being included in a symposium of Northwest lichenologists. So my goal here is in the next 12 minutes to tell you a little bit about work that I'm doing both together with Jessie Miller and undergraduate students to leverage biological collections in undergraduate research and map the distribution of a host and a parasite in a lichen system. And so what I'd like to start with here, and let me move this out of the way here. I'd like to begin with a quote that has really stayed with me since graduate school, and which I think probably resonates with everyone here. And it goes like this: for animals as well as plants, there have never been individuals. We are all lichens. And as an ecologist who happens to work with lichens, more and more as I'm building a research program that is accessible to student researchers, I keep coming back to this idea that symbionts really are ubiquitous and can provide a useful framework for studying diverse aspects of ecology. So we learn in an introductory ecology class that symbionts range from parasitic, like this apicomplexan that causes malaria, to mutualistic, such as the famous symbiodinium dinoflagellate that corals have to have in order to live, to somewhere in between on this spectrum. But we also know that symbionts don't always live where their hosts do. So for example, Plasmodium here is restricted to tropical and subtropical areas, predominantly in Africa. Yet both humans and the Anopheles mosquitoes that host this parasite live in places where Plasmodium doesn't occur. And similarly, research shows that Symbiodinium clades and species are found in different geographic regions of the world. Okay. So we can ask generally, what is it that limits a symbiont's distribution? And as an ecologist, usually we focus on some set of these three overlapping factors: host traits, environment, and transmission dynamics. So for example, the Australian Institute of Marine Sciences has several research programs aimed at saving corals from the effects of ocean warming. As many of you know, warming events disrupt this symbiosis, bleaching corals and eventually killing them. And so they've really focused on thinking about artificially selecting warm-tolerant strains of the symbiont to introduce tolerant strains into the wild and maintain the current distribution of corals. On the flip side, we're really interested in limiting the distribution of the malaria parasite. And so research on this is focused on efforts to limit mosquito vector transmission, such as through bed nets and removing environments where mosquitoes breed. What I'm going to use this framework for is to think about what factors limit the distribution of a lichen symbiont at two different spatial scales. So specifically, we're going to be using field research by undergraduates to explore how host traits and population density influences the local prevalence of a particular symbiont here in Northern California. And we'll also be examining how environmental factors may limit the geographic range of this symbiont relative to its host using herbarium collections from the entire Western United States. And so if you haven't guessed, your host for today here is the lovely ubiquitous Evernia prunastri, and the star of the show is a little lichenicolous fungus named Unguiculariopsis lataui. Please correct my pronunciation later. I've never actually heard someone say this name before. And so because of its unwieldy name, my colleague Jessie Miller and his students have dubbed this little lichenal Little Ung or Young Larry. And I'll go with Little Ung for this talk. And we really like this system for four main reasons. First, this host, Evernia prunastri, is pretty hard to misidentify, and therefore is a good candidate for student research. It's really abundant, but the population density varies widely across our region, as does, it looks like, the infection prevalence by this symbiont. The symbiont here has a really unique appearance. It has this it produces these blast gas stomata on the surface of the lichen thallus. If you look at them closely, they look a little powdery, but actually those are cortical hairs that are sort of diagnostic of the species. And it's easily found even without a hand lens. You can see this individual here just heavily infected by this symbiont, which we think may be parasitic. We haven't decided that yet. That's another project. And the other reason we like this system is if you look, these two individuals are about the same size and age. They're living adjacent to one another on the same substrate, and yet one individual appears uninfected and the other is heavily infected. And so individuals vary widely in infection severity. So that's a really interesting question to examine. So let's start by thinking about what potentially limits the local prevalence of this little ung in a population of Evernia. We think that this little lichenol spreads through spores that are released by the ascomata it produces on the surface of its host, thereby spreading to nearby uninfected host. And so we can come up with this very basic prediction from ecology that the prevalence of the infection in the population should be strongly related to the population density of the host. And that the density of the host is probably determined by a mixture of climatic factors as well as air pollution, since it is a lichen after all. However, we don't necessarily hypothesize a direct relationship between environmental factors and the prevalence of little ung. And so over the last 2 summers, I've had 2 students at St. Mary's summer research program, Dante Martin pictured here and Alex Hildenbrand. They surveyed 13 oak woodland sites here in Northern California for both Evernia abundance and the prevalence of little ung in the population. And we were surprised to find fairly high infection rates with up to 75% of individuals infected in one population here. We also see a fairly strong relationship between Evernia abundance, if you look at sort of per meter canopy branch length, and the prevalence of little ung, which is consistent with a primarily horizontal mode of transmission here. When we took a similar data set from my colleague, Jesse Miller, who has been sort of teaching an undergraduate ecology research course there for 2 years, even though they sampled in a different and more restricted geographic region and only surveyed 10 individuals per site, we saw a really consistent relationship here. But the question is, does this relationship hold true at larger spatial scales? Is it primarily host abundance that is limiting symbiont prevalence? Is it really that transmission dynamics here are possibly the primary limiter of the distribution of this symbiont? And so Evernia prunastri has a global distribution occurring in Western North America and in Europe, not here in Eastern North America, thanks GBIF. So here's the location of our surveys in California. And if we focus just on the range of the host here in Northern North America, the question I wanted to answer is the range of little ung here restricted relative to its host, or is it just a function of the abundance of its host? Are there environmental factors that limit the distribution of little ung beyond that of their effect in determining the abundance of Evernia? Okay, so we sort of have 2 pathways here, climate and air pollution limiting Evernia abundance, or some additional direct effect on the prevalence of this symbiont. So here's what we did. I want to go back a second. So to examine this hypothesis, I utilized a really fantastic resource that's recently been compiled by Sarah Jovan and colleagues at the US Forest Service. She has compiled all of the Forest Service's previous lichen surveys into an aggregate database that she really generously gave me early access to. And so as many of you are aware, these data are unique because they use a standard survey protocol to estimate lichen species abundance on forested sites across the US, with a very heavy concentration here in the Pacific Northwest. And so what I'm showing here is points which are colored by their estimated Evernia abundance, with gray points showing survey locations where Evernia was absent. Okay. And what we did with this data was in 2019, I went up to Oregon State University where the physical collections from this database are housed, and I examined about 720 of these sites to determine whether little ung is present. And so what's shown here is the abundance of Evernia just at the sites where it's present. And then I examined them and I decided whether little ung is detected or not detected at each of those sites. And so I detected little ung at about 37 of these sites. And so now what we have is one data set that we can use to test the relationship between environment, Evernia abundance, and the prevalence, or not the prevalence, but just the presence of little ung. And certainly there's an issue with detection probability. I'm guessing based on some other work, I think that the probability that I detect little ung from a single survey year here is probably less than 30%, but greater than, sorry, less than 50, but probably greater than 30% here. So it does show up in these collections. So what did I do? I took this data set, and first I modeled the occurrence of Evernia in the region using a combination of long-term average air pollution data from the National Atmospheric Deposition Program and climate variables from PRISM. So what's showing here is a predicted presence of Evernia from this model using these abiotic variables. And what you can see here, actually, one other thing was included. I included ecoregion as a categorical predictor just to adjust for biogeographic differences in Evernia presence, which aren't captured by sort of abiotic variables here. And so this sort of predicted distribution of Evernia from the model really does capture what we know about the actual distribution of the species outside of this data set. It has pretty decent predictive power here, according to cross-validation metrics. So I took that model, and so we have an estimated distribution of Evernia. And now what I wanted to know is how well does the presence of Evernia predict the presence of little ung? Strictly from the model that we've already used to predict Evernia? Or do I need to add in additional air pollution and climate predictors to understand the distribution of little ung in this region, in addition to just knowing whether Evernia is present or not and its abundance? Okay. So here's the model predicting the occurrence of little ung. And based on this map, it appears that the range of little ung is restricted to the western part of Evernia's range, and really not as frequent in parts of its northern parts of its host range. If you look at individual predictors in this model, I'm going to just show you data from as if we're in the Mediterranean, California ecoregion. What I'm showing here is the sort of predicted occurrence from the model of the host, Evernia prunastri, alongside the predicted occurrence of little ung. And so remember that the prediction for little ung also already includes the distribution of its host. And so it appears that Evernia might have a slightly lower cold tolerance, so it's less likely to occur at lower minimum temperatures than its host. But that really, if you look at this blue line, it doesn't have any additional response to sort of maximum temperature or humidity. All of its response to those two variables can be found from the distribution of its host. But I think one of the most interesting relationships here is that little ung does actually appear to occur in areas with greater nitrogen deposition. I'm wondering if maybe this suggests the parasite is nitrogen limited, and that agricultural air pollution could facilitate its spread. Or with any sort of correlative modeling approach here, there could be some other covariate that's not getting captured. So that's just a kind of very initial hypothesis. But this really, I want to emphasize, this is really still a work in progress. I'm not quite ready to trust this model. So with the help of several colleagues, I've looked at 427 other herbarium collections of Evernia. We've found little ung on 111 of them. 35 of these have geographic coordinates, which are shown as pluses. And what you can really see is that the model is doing a really poor job of predicting the occurrence of little ung in Southern California. And I believe that's just because the model is trained on a data set that's so heavily biased towards the Pacific Northwest. And so the way forward here is really to build some type of joint distribution model that utilizes the survey design of the Forest Service data balanced with the broader geographic scope of sort of haphazard herbarium sampling. And I'd love to get suggestions on this if anybody wants to chat later. So to conclude, we've seen that local prevalence is strongly related to host abundance, probably because of transmission dynamics. We've seen that the northern geographic range of this potential parasite might be a bit more restricted than its host, possibly due to temperature limitation, possibly due to biogeography. But to me, really, the most exciting thing about this work is that there's just so much space here for novel student research because we have so many open questions to answer about the interactions between this fungal symbiont and its environment. So to wrap up, thank you so much for listening. Really thank all of these participants in our research here, as well as the parks that allowed us to collect lichens. Particularly grateful to the wonderful summer research program that we have at St. Mary's that funded 2 students on this project. And I have 1 minute to make a quick plug from my other role as co-editor of Bulletin of the California Lichen Society. If you're doing work on lichens or taking pictures of lichens that would be interest to our members in California, I really encourage you to send your contributions to myself and Justin Schaefer at editor@californialichens.org. COVID's kind of slowed down our production of the 2020 issues, but we're still actively looking for submissions for the 2021 summer issue. So we accept technical articles, non-technical articles, caption photos, news, short submissions, things like that. So thank you so much for listening, and I look forward to hearing your questions and ideas. We'll go back to that.
Thank you, Jess. That was wonderful. We have a couple of questions in the chat. We have time, Meaghan?
Yeah, definitely for 2 or 3 questions, I think.
Great. So Heather was wondering about the distribution of the photobiont in Ivernia prunastri being kind of uneven. So we have here, the photobiont can traverse from one surface to another depending on how the thallus is oriented to the sun. Does this trait make it more vulnerable to auxiliary fungal symbionts?
I have no idea. That's really interesting. One of the things I like about this system is there's so much room for projects on how host traits like that, the distribution of the photobiont might affect infection severity or probability. And so I definitely want to pursue projects like that. I have no idea how to answer that question.
That's a great question. Okay, and so John is wondering, how do you account for if little ung was present but not on the thallus collected in the plot, especially if it was at a lower abundance in that plot?
Yeah, that's not accounted for. So basically this data set is biased towards non-detection because they only collect a couple individuals at a time. And so certainly we're going to see somewhat of a bias here between low Ivernia abundance and low probability of collecting little ung, which could diminish the correlation between those 2 variables. And as I said, this is a work in progress, and so I'd be interested in a little bit more sophisticated modeling techniques that could incorporate that lack of that detection probability.
Great, thank you. Well, we're at 11:50. I think there are a couple more questions in the chat that you might want to take a look at. And thank you again for your wonderful talk. That was great.
Thank you so much. I appreciate it.